So in my lab, we're interested in understanding, controlling, and designing microbial communities. So in a sense, we're interested in very different types of microbiomes. So here are three examples that you see on this picture. So on the first one shows the image of a gut community. So this actually comes from the honeybee gut, where different species are labeled so that we can see sort of where the bee finishes and where the microbiome starts, and sort of how the interactions take place between these members. Another system that's really interesting to us is the soil microbiome. So where different species, many different species, can live in the same place and can shape the environment and also contribute to the productivity of the plants that are growing in the soil. But we're also interested in more human-made microbiomes. For example, the ones that you see in this picture are showing the rind of cheese. And of course, it's the microbiome that lives on the surface of this cheese that actually contributes to the flavor that you would get. And so the idea here is that we don't actually work on any of these three systems, but we are interested generally in understanding what are the commonalities between these systems, how do we overall understand the ecology and evolution of microbiomes. And if we can understand it, the hope is that also we can control these systems to our advantage. So whatever that might be, maybe it's soil health, or it's the bee health, or it's a different flavor of cheese, or we can even design new microbial communities from scratch. And so to do this in my lab, we combine both experimental systems, and we also build mathematical and computational models. And the idea again is to study these general principles. So even though we're a general principles lab, we also always like to have sort of a target application in mind that helps to kind of guide our research towards something useful. So here are a couple of examples. So one would be if you could build a community from scratch. So you would choose certain members of different microbial species and put them together to convert greenhouse gases into useful chemicals. So we know that there are many microbes that feed on greenhouse gases, whether they're CO2 or nitrous oxide or methane. And then we also know that there are microbes that produce the building blocks that we might use in a lot of our products. So whether they're bioplastics, for example, or other different chemicals. And so if we were able to understand the general principles that allow a community, for example, to be stable, so we would like different species to coexist over a long time scale and retain the functions that we're interested in, then one could have potentially very important applications. Another potential application that's maybe closer to what people working on microbiomes might think about is whether we can drive the microbiome of the skin towards a different state, which would actually repel mosquitoes. So we know, and you can see this in these images on the slide. So here, for example, this graph is showing different attractivity of people to mosquitoes. So we know that some people are really attractive, and ideally you want to be sitting next to one of them so that you get protected against the mosquitoes. But what makes them attractive is actually the partly body heat and CO2, but the body odors that are produced by the microbiome that's living on the skin. So that suggests that if you could make a probiotic cream that would contain the right species combination to actually have a microbiome that does more what's on the left hand of this plot, so more repelling the mosquitoes, then that could actually have very important medical applications. So with these applications in mind, then how do we go out to study the general principles that allow us to get there? So obviously studying a single microbial species is too simple, but for us we also find the whole microbiome in its natural state in the environment too complex. And so in my lab what we do is we bring these communities into the lab and we simplify. So we take a handful of species, maybe say on average about 10 species, and then the idea is that with a few of these species, then you can work out all the interactions that are happening between them and try and predict what will happen if you remove one species or if you change the environment, and how the whole system will evolve over time. So broadly, I could say that our group works on these 5 different topics. So we're interested in interspecies interactions. So those were the arrows that you saw on the previous slide. We're interested in controlling community dynamics. So who exists, who goes extinct. You'll see a little bit of this today. We're interested in the mapping between community composition and community function. So how do you get a specific function from your community? How do we predict that, and can we optimize it as well? We also look at not just well-mixed microbial communities, but ones that are organized in space. And we're interested both in the ecology, but also in the evolution that happens within these communities. And today I'll be talking about the two topics that are in bold, so interspecies interactions. And if I have time, I'll have to keep an eye on the time. If there's time, then I'll also briefly talk about predicting and optimizing community function. Then if you're interested, the QR code will take you to our website. Okay, so the first topic, as I said, will be about interspecies interactions. And today I'll be talking briefly about the work of many different people, most of whom have already left the lab. So everybody except Clément is no longer in the lab and either did their PhD or their postdoc work within the lab. Okay, so why do we first need to study these interspecies interactions? So if we look at any of these systems here and imagine that we want to predict their community dynamics. So we want to predict which of the species, for example, in the bee gut might persist in the long term or which ones might go extinct, then we need to understand how it's interacting with the species that share the same environment. Are they competing? Are they maybe feeding and relying on each other? Or how does the system work? So we set out to understand better these interactions. So just for us all to be on the same page, I'd like to define what I mean by interactions. And there it's the effect of one species on the growth and death of another. So a very classic example comes from predator and prey relationships, where the prey experiences negative effects on its growth or positive effect on its death. Let's formulate it in terms of negative effects on the population size. And then, of course, the presence of the prey has a positive effect on the predator. And so historically, we've looked from ecology as a field has looked at these different interactions kind of in their trophic levels. So you always have these predators and the prey might be predators of another layer below it. And a lot of the theory that's been developed in ecology has been based on this view. But then if we start to look at microbial ecology, which is a younger field, then things became a little bit more messy, especially if we look at interactions between bacteria that are maybe within the same trophic level, more or less. And then the interactions are based on different things. So maybe there's one species that provides food for another or kills a different species. Sometimes they make biofilms that might protect each other. So the relationships seem not as obvious or not as structured as they might be, or at least the way we used to think about interactions between species. So then the question in the field kind of arose, how do microbial species interact? Do they tend to sort of help each other typically and feed each other, or is there a lot of competition between them? And so the field sort of diverged into two different directions. So there were several people more on the competition side of things. So you see here two titles of papers from not so long ago. So competition, not cooperation, dominates interactions amongst culturable species. Or this one here, bacterial species rarely work together. This was an opinion piece. And then sort of the other side of the coin, there were people saying kind of the opposite. So microbial positive interactions are common among culturable bacteria, which seems almost the opposite title to this one here, or metabolic exchanges are ubiquitous in natural communities. And if you sort of follow these discussions a little bit on social media or at a conference, they can actually get quite heated. And so it seemed like we needed some clarity in the field. So when I started my lab, the idea was to set up a model system where we could ask these questions and test, okay, which types of interactions do we see under which contexts? So we set up this model system, this experimental system, which are 4 bacterial species that you see listed here. And these were actually isolated and are used to degrade pollutants from so-called metalworking fluids. So metalworking fluids are machine oils that are used in factories that do any metal cutting, and have large machines that need cooling and lubricating. And so that's what these oils do. And you would use them in many, many liters within a factory. And then once they have expired, then you need to sort of throw them out. And so obviously you cannot just pour this into the river, but you need to treat the liquid first. So you could do this chemically, or what they found in this patent, so this was before I started working on this system, they actually It was these authors that isolated these first 4 species and showed that they were capable of reducing the pollution load of these machine oils. And so this is what you see in this photo here. So this is from my own lab, where you can see these machine oils. So they're typically a oil and water emulsion. That's why it looks milky. And then if you grow the bacteria in that liquid for about a week, it can reduce the pollution load. And so for us, this was a nice system because we have a limited number of species. Again, we just have 4. We can work out all the interactions between them. And we also have a function. So we can measure what is the effect of adding or removing species on the function of the system. So this is what Philippe started out with as his first project. And the idea there was to measure the interactions between these 4. So thinking of the interaction definition that I had, so how does each species affect the population size of the other? So if we take this species here, this green one, AT, and then we'll grow it in cultures either alone or together with each of the partner species, so with OA, CT, or MS. And then we want to see how does its growth differ between the monoculture and the co-culture. So that's what you see in this example plot here. So the green lines are AT when it's alone, and then the different black lines are AT with each of the different partners. And you can see that the growth patterns do change over time. And this is just a batch culture with leaving the species to grow for 12 days. Okay, so when you have this kind of data, so you could do this not just for AT, of course, but also for each of the other 4 species. And then we can sort of compile a network of interactions. And so, for example, here, you might, just by eye already, you can tell that when CT is present, AT grows much better. So that would be called a positive interaction. So we would draw a green arrow, as you can see here. So green means positive between going from CT to AT, meaning CT helps AT to grow better. And then we did this for all of the other possible interactions. A negative would have been in brown, but we actually don't see any negative interactions in our system. And here the thickness of the arrow is the fold change in population size in the presence of another species. Okay, so we found only positive interactions. And this was a bit surprising for me, at least, because I had done my postdoc in one of the labs that was on the camp that said it's mostly competition that you should see between microbes. And so Philippe started looking in the literature, and he found this concept that comes from plant ecology called the stress gradient hypothesis. And the idea there is if species are growing in a stressful environment, so originally plants are growing in a stressful environment, they tend to benefit more from other species that are around them. So you would be more likely to see positive interactions. And so Björn was another PhD student in the group who was a theoretician. And so he built a model to try and understand this. And the hypothesis that we were trying to test was that maybe the 4 species in our system, they interact positively because the machine oil is actually toxic. And this is something that we knew from the start, and you can see it if I go back to the previous slide here, that AT is sometimes dying in the medium, and there are actually 2 other species that would also die completely if they're by themselves. And so that's because when they design the machine oil, they put antimicrobials in there to keep them sterile. Okay, so why would it be that the toxicity would favor positive interactions? So Björn's model was really helpful with that, and I won't show you details with equations, but actually Philippe, who is not a modeler, decided to make an animation that explained Björn's model that I hope you will find helpful. So the idea is as follows. If you have one bacterial species that's growing in monoculture in an environment that has just nutrients, it will consume the nutrients, which will allow it to grow, and then you will get a larger population size. Now imagine you take the same species, but you add a second species to the same culture that also eats the same nutrients, then they will compete for it, and you'll end up with a smaller population size of the red one. So then by comparing co-culture and monoculture, you can say, okay, there's competition or negative interaction from the green to the red because they're competing for the nutrient source. Now imagine your environment is a bit different. It's not just full of nutrients, but it also has toxins. And these toxins kill the red species. Then it'll die completely if it's by itself. But imagine now that the green species actually reduces the toxin, so it degrades the toxin that's in the environment. And in this case, the red species will be able to grow. So if you actually don't know what's in your environment and you happen to grow your species in an environment that's like the one below, then you will be comparing monoculture to co-culture and you'll say, oh, there's a positive interaction. But what you won't be able to see is that there's also a negative interaction happening between these species, but you never measure it because your environment always has toxins in it. Okay, so then the prediction was if we could take the original machine oil and make it less stressful. So the idea was ideally we would remove the toxicity from it, but that's very hard to do because you just buy the machine oil off the shelf. So instead we thought maybe we can make it more benign by adding amino acids, which the microbes would eat, or we could just remove the machine oil completely and just put amino acids. So the idea is here we're changing the balance between toxicity and nutrients. Then the hypothesis would be that with less environmental stress, we should see more competition. And indeed, that is what we saw. So when we did all the measurements again of all of the interactions, you can see now these brown arrows appearing, which are indicative of competition. And it turns out that the species that compete are the ones that are able to grow. So here, when you add amino acids, AT is able to grow, and then it starts to compete with CT. So the conclusion from these experiments was that reducing environmental stress can increase negative interactions. And that's because more species are able to grow alone, and as soon as species can grow alone, then they start to compete with one another. Okay, so that was all quite nice, but again, it's relatively fundamental and maybe a bit, not mechanistic. So what we wanted to do next was to see if we can simplify the system a little bit, that we can come to a mechanism behind this phenomenon. And so Rita was a PhD student in the lab, and Aurore was a postdoc, again, an experimentalist theoretician pair. And the idea there was to test the stress gradient hypothesis, but with just two species, just to keep it a bit simpler, and then rather than the complicated machine oil, have a single compound that plays the role both of a nutrient and a toxin. And so Rita chose linoleic acid, which is a compound that's often used in machine oils. And when you put this compound at a low concentration, it acts as a nutrient for both of these two species. So we just picked out these two species from our 4-species community. But then if you increase the concentration of linoleic acid, it becomes toxic for AT, but is still a nutrient for CT. And so the prediction then was that at low concentration, when it's a nutrient for both, we should see competition. But then when it becomes toxic for one species, we would maybe see a positive effect of CT on AT. So that's indeed what we saw. So here are the data. So I'm showing you again these growth curves over 8 days in this case. The solid lines show the species alone, and the dashed lines show them in co-culture. So what you can see here, if you look at just the green, so that's AT alone, and then AT in the presence of CT, it grows worse. And this is at low concentration of linoleic acid when it's a nutrient for both. Now, if you increase the concentration, now it becomes toxic for AT, and that you can see because the solid line, so the monoculture of AT, is dying. But then when it's in co-culture, when CT is present, then it grows fine. So basically, we can say that at low competition, at low concentration of linoleic acid, we have competition, and at high concentration, we have facilitation or positive effect from CT to AT. So that means that by simply changing the concentration of one compound, we can change the interaction sign from negative to positive. Now, could we also go the other way around? So for that, we would need to understand what's actually generating this toxicity. And Rita discovered that over time, linoleic acid accumulates reactive oxygen species. And that has nothing to do with the microbes. That's simply just the chemistry of the molecule, that it reacts with oxygen, and then you get these reactive oxygen species in the environment. And that's what's toxic for AT. So that's what's killing it. And it seems that CT is able to remove these reactive oxygen species, and we could measure that in experiments. And that's what is responsible for the facilitation from CT to AT. And so we thought, what if we actually remove the reactive oxygen species ourselves? And so Rita did that using an antioxidant. So she added that from the environment, and the idea was then she would break this facilitation between them, and then perhaps we would just be left with competition. So looking at the data, so now I'm only showing you AT, but CT is still in the culture. but you can see a repeat of the previous experiment. So again, AT dies by itself and then grows fine when CT is present, so in the co-culture. And now in the second plot, we're looking at the same situation, so high linoleic acid concentration, but we also add TBHQ, which is an antioxidant that removes reactive oxygen species. And now what you see is that they still grow fine in co-culture, but they grow even better if they're alone, meaning that there's still this competitive effect that's there, but now by adding the antioxidant, we were able to unmask the competition, and now we're able to reveal it. So removing the reactive oxygen species restores competition at high linoleic acid concentration. So we could really go back and forth between these. Okay, so back to the debate on the fields. Our position is essentially, it depends. Okay, so you don't always get competition, you don't always get cooperation. It depends on the environment that species are in. And probably what's happening is typically between two species, there will be several levels of interaction that are happening at the same time. And depending on if your environment is more toxic or has more nutrients, that can change the net interaction that you would measure. So during this time, Oliver Meacock was a postdoc who arrived in the lab, and he was seeing all of this unfold. And he is a theoretician, and he thought, I'm going to build a predictive model that will help us tell also under which conditions we might expect competition or cooperation. And so he built a model that's even simpler than Björn's. So this was the diagram illustrating Björn's, and what Ollie did was to even remove the second species and just think about a single species in an environment that has nutrients and toxins and how that species interacts with itself. So just as the simplest case that he could think of. And what his model was able to show was that you can decompose the interaction with a species with itself into two components. So the first component is how the species affects the environment. So you can imagine that this environment we can plot the environmental states on two axes. One axis is the concentration of the nutrient, and the other axis is the concentration of the toxin. And these little arrows in the plot show in which direction the environment will go as this green species is growing. So this green species can degrade the toxin. So you can imagine that maybe the toxin is an antibiotic and the green species is resistant to it, so it can degrade the antibiotic. And there's also a food that, of course, it takes up. And so as it grows, then the whole system is going towards the 0-0 point where there's no more toxin and no more nutrient left. At the same time, the flip side is to look at how the environment affects the species growth. So in this little plot here, what you can see is, again, if you look at different nutrient concentrations and different toxin concentrations, the colors show the growth rate of the green species, and the little arrows show kind of where it wants to go. So ideally, it would go to an environment that has lots of nutrients and very few toxins. Okay, and that's where its growth rate would be maximized. And then the trick that Ollie discovered is that if you just multiply these two by each other, what you get is a map of so-called instantaneous interactions, meaning that depending on where you are in this space, we can predict what is the interaction that will be there at that location. So in this plot, the green color means that the interaction will be positive. So if you have high toxicity and high nutrients, then you would expect a positive interaction. And then if you're down here at very low toxin concentration and low nutrients, then you expect more competition between species. So instantaneous means that you just have to look at the interaction that would be happening given the context in a moment of time. So if we now consider an example, so a concrete example of, say, this bacterium growing in a batch culture with some nutrients and an antibiotic, what we need to think about is that over time, as it grows in the batch culture, the environment is changing, right? Because if you start from this red dot here, so this would be the initial nutrient concentration and initial toxin concentration, then as the microbes grow, then they consume the nutrients and degrade the toxins, and they take the environment across this trajectory towards the 0-0 point. And that means that throughout your batch culture, the interactions are changing. So at the beginning of your batch culture, they're maybe facilitating each other, but by the end, they're competing. And we can plot that on a diagram. So this yellow line here is just a 2D projection this color here. So they start out with a positive interaction, and then the interaction becomes competitive after some time. And of course, this depends on where you start, so which concentration you start with. And what you see in blue is the cumulative interaction. So if you were to stop the experiment at any point in time, and you compare the monoculture and co-culture, I suppose, which is a bit of a detail here, but I can explain it in the questions time if you want, then we would see this kind of interaction. So at the beginning, the environment is dominated by this detoxification part, meaning that the interaction is mostly positive, because the more the species removes the toxin, the more it can grow. And then in the later part of the batch culture, there's competition for nutrients, because the toxin is all gone, and so the only thing that's left are the nutrients that the species are competing for. And so what Ollie could then do with this framework was not just look at the single species, but he applied it to some experimental work that he did himself. And he also took 2 published studies, one that had batch culture experiments with 2 species that were sort of degrading different compounds and taking up different compounds. And then the second study that was more spatial. And then he could show that in both cases, he could predict how the interactions would change over time. So as the environment traveled across the space, going from more cooperation to more competition. And this works whether it's time or space. So that he could show that this was quite general. So this is really nice, because it means that we have now a framework with which we can really, assuming that we understand how each microbe changes the environment and how the environment changes the growth of each of the species, that by putting all of these together into one system, we should be able to predict how the interactions will change over time or over space. Okay, so moving on now to a more applied side of this work. So we had a follow-up project with Andrea, who is a former PhD student, and Clément, who's currently a senior scientist in the lab. And their idea was to see if we can actually use these concepts to control species abundances in a useful way. So when they're thinking useful, they were actually thinking about pathogens and whether we could use this understanding to suppress pathogen growth. And so essentially we have the same type of diagram as before, but now we can put different identities on the species. So we could take here a species that's a pathogen. So in our case, we took Pseudomonas aeruginosa strain PA14, if you're familiar with it. And then we took a second species here called Citrobacter freundii, which is not exactly a commensal, so that's why it's in quotation marks. But for the purpose of this proof of concept, we can think of it as a commensal. And we picked these species because we knew from some preliminary experiments that they would compete for a carbon source, which in our case was glucose. But then also, as they consume glucose, Citrobacter tends to acidify the environment quite strongly, and this acidification inhibits Pseudomonas aeruginosa. So we thought, okay, if you wanted to inhibit Pseudomonas aeruginosa, which is a pathogen, could you then increase the nutrient concentration such that Citrobacter freundii would acidify the environment even more, killing Pseudomonas aeruginosa? So that was the principle, and we tested this in the lab. So what I'm showing you here are growth curves again, this time transfers, but it's a bit of a detail, it doesn't really matter. In essence, the x-axis is time. We have a co-culture of Citrobacter and Pseudomonas, but what I'm plotting is only the population size of Pseudomonas. Okay, so only the red species, and each column has a different glucose concentration. So we start with 15, then 30, and then 45. And what you see in the bottom row here is the pH of the environment. So you can see that as you increase the glucose concentration, there's more and more acidification. And indeed, what we see if we look at the population size of Pseudomonas is that it drops more and more over time. We had actually, in this experiment, also applied some antibiotics. But in essence, you see that regardless of the antibiotic concentration, if the glucose concentration is high enough, then Citrobacter can completely eliminate Pseudomonas aeruginosa even without the antibiotics. Okay, so essentially this is just a way to illustrate how this framework can help us to actually think of applications. So concretely here, the idea would be to feed a probiotic strain to make it more effective against the pathogen. And we also, in this paper, we tested several pathogens. So in addition to Pseudomonas aeruginosa, we tried with Klebsiella pneumoniae, with Agrobacterium tumefaciens, and with both of these, the principle works. And we also replaced our commensal with another one, which is a common probiotic called Lactobacillus plantarum. Okay, so I'll just summarize this part. So essentially what I've shown you, so the work of all of these people, is that environmental toxicity favors positive interactions. So this was this idea of the stress gradient hypothesis. And the reason why this works is because when a species cannot grow, it's likely to be facilitated by another species that can grow. And then if you change the environment, you can change the interaction sign. So you can go back and forth between cooperation or facilitation and competition, which was the work of Rita and Aurore. And then also that these changes can be predictable in time and space, and that we've built this this model, this theoretical framework by Ollie that can show how one can do this. And then using this logic, we can actually apply it also to useful things like eliminating a pathogen. So I see I'm almost out of time, but I might take just 5 minutes to quickly tell you about the second project. So the second topic that I wanted to talk about was optimizing community function, because so far we've just talked about the interactions between the different species, but of course they also have a function, so some utility. And in the machine oil example that I told you about before, the idea was to reduce the pollutant load. So that's what you see in this plot here. You see each of the species alone cannot really reduce the pollutant so much, but if you put all four together, they can work well together to reduce the pollutant. However, there's still quite a bit of pollutant remaining. Okay, so the y-axis actually doesn't start at 0, so we end up with around 5, when ideally we would be able to remove all the toxicity. So the question at this point was, okay, why did we choose these 4 species? So they were actually not chosen by us, but by former collaborators, and they had isolated them from the environment and just found, okay, these 4 work. But what if we would take a different combination of 4? And how do we search through the space of all possible combinations of species? So the idea that we had was to do that using a breeding approach, so artificial selection approach. So the idea is that you compete lots of different communities against each other, and then you always pick the best ones, and then again and again favor those, and over time, hopefully, you would find the right combination of species that performs the best. And we're not the only ones to try this, so there's quite a bit of literature, and here's just the state of the art until 2022. There are also more recent papers, but it actually turns out it's very difficult to do with whole communities or whole microbiomes. So the principle is as follows. So you take some communities, so composed of a few species, you let them grow, you incubate them, you measure whatever trait it is. So in our case, it would be pollutant degradation. You rank them all by who does best from worst to best, and you take the best ones, and you start a new round of selection with those best ones. And so our innovation was to rethink a bit this step number 4, this redistribution. So how do you create offspring communities from the parent, the most successful parent communities? And our approach was to disassemble the communities. So essentially to take out the winning species from each of these communities and then rebuild new communities that either were identical to the previous round's winning communities or had some mutations in species composition, meaning that sometimes we threw out a species, sometimes we added a species or exchanged a species, etc. And that way we could explore the space of possible species combinations. And we did this both using a model, so that's in this paper here, and then we also did some experiments which were quite extensive. So I'll just show you a sneak peek into these experiments. You can take a look at the paper if you like. And essentially the difficulty was actually disassembling these communities after every round. So if you know much about growing species, microbial species in the lab, this meant making selective media for each of the different species such that we would be able to separate them and then recombine them in the next round. And we did this experiment for 18 rounds and also with a random control. And I'll just show you one quick results slide. So in the top part of the plot, you see the selection treatment, and in the bottom, we're just selecting randomly, so to contrast, just as a control. And then on the x-axis, there's all of the communities we tested ranked from best to worst, from worst on the left to best on the right. And the only thing you really need to see is that in the selection treatment, you get this accumulation of dots that is sort of going up, so improving in their score as the weeks go by, where the weeks are on the y-axis. So essentially what this allowed us to do was to find a community with a different species combination that's 70% better than the original community. So that was quite nice. It's not huge. So of course, ideally we would want it to be 10 times better than the original community, but at least we do see some improvement. So at the moment, what we're doing is revising this method and also trying to make it a bit less cumbersome with having to isolate all the species every single time. And we're currently testing this disassembly 2.0 on 2 different systems in collaboration with Itzik Mizrahi's lab, and Sarah Winkler is doing the experiments. And Afra from my lab is doing the theory. And then we're also testing that, so this here, the system is to maximize fiber degradation of from a cow microbiome. And here the idea is to use a microbiome to kill viruses in lake water. And both of these things are things we can optimize with our algorithm. Okay, so that brings me to the end of the talk. So just a couple of take-home messages. So I've shown you in the first part that changing the environment can change interactions between species. So just remember, if you're going to measure interactions between microbial species in a given community, these will not be constant. This will very much depend on how you grow them, how you measure the interactions, at what point in time or in space you measure the interactions. And you can also use that to control them. And then the second take-home message is that we can actually optimize bacterial community functions even blindly. So without understanding the mechanism of exactly how species are interacting or what they're doing in terms of the function, but simply by breeding them towards an optimum. And my last take-home message is it really takes a small village. So I've shown you many, many pictures of the people who've worked really hard to do all of these experiments and build all of these models. And I'm very thankful to them, which brings me to the acknowledgement slide, particularly of the current lab members that you see in this photo, many of the former lab members whose work I presented today, collaborators, and the funding bodies, and also the taxpayers behind the funding bodies. And thank you for your attention.